Hi everyone, today we'll be talking about real-time polymerase chain reaction, also known as real-time PCR. My name is Sarai Mian and this is...
I'm Sebastian Mesa.
And I'm Valeria Gonzalez.
And the objective for today is for students to familiarize themselves with the principle of real-time PCR, the steps of real-time PCR, and how they are all related.
We will also be talking about the different applications for this technique and how they have evolved for today's use.
Traditional PCR has evolved into real-time PCR, which is more commonly used. In this graph, we have the cell cycle on the x-axis and the DNA concentration on the y. Real-time PCR detects DNA amplification early on in the stages of the reaction, as opposed to traditional PCR, which detects it at the end point. Real-time PCR uses a fluorescent probe to detect this amplification early on in the stages. In the graph, we can see that as the cell cycle increases along the x-axis, the DNA concentration increases exponentially until it reaches its plateau, where this is where traditional PCR detects it at its endpoint.
Real-time PCR is a PCR-based technique that is simultaneously amplifying and detecting changes in the concentration. Real-time PCR collects data during PCR amplification by utilizing fluorescent signals emitted by special probes or DNA dyeing dyes. To understand real-time PCR, it is easier to start off with the basics of PCR. PCR is a technique used to amplify DNA. A thermostable polymerase enzyme drives PCR. This PCR will synthesize a complementary sequence of bases to any single strand of DNA, providing it with a double-stranded DNA. This is useful because you can choose which gene you want to polymerize. In a mixed DNA sample, you can add small tiny pieces of DNA to find a complementary DNA to a gene. These small pieces are known as primers because they prime to DNA and make a sample ready for the polymerase to bind. During a PCR, changes in temperature are used to control the activity of the polymerase binding to the primers. The first step is denaturation. To begin the reaction, the temperature is raised to 98 degrees Celsius. At this temperature, all double-stranded DNA are melted into single strands. The second step is primer annealing. The temperature is then lowered to about 60 degrees Celsius, allowing the primers to bind to the gene of interest. Thus, the polymerase has somewhere to bind and can begin to copy the DNA strand. The third step is extension. The optimal temperature for the polymerase to operate is 72 degrees Celsius. At this point, the temperature allows the enzyme to work faster. Now, there are twice as many copies as there was before. The temperature is changed repeatedly for about 40 cycles. 1 copy becomes 2, 2 becomes 4, 4 becomes 8, until there are billions of copies that are created. After amplifying the gene, it is possible to amplify DNA out onto an agarose gel that is stained with a dye that makes it visible. The brighter the band, the more copies of your target gene that have been created. The same principles of amplification are used in real-time PCR, but instead of looking at bands on a gel at the end of the reaction, the process is monitored in real time. Literally, the reaction is placed into a real-time PCR machine that watches the reaction occur with a camera or a detector. There are many different techniques that are used to allow this process in a PCR reaction to be monitored. They all have one thing in common though. They can link the amplification of gene to the generation of fluorescence, which can be simply detected on a camera during each PCR sample.
Before we get into the procedure of real-time PCR, let me give you some background and introduce our materials that we'll be using. In order to get reliable and accurate results, proper instrumentation is required. The laboratory exercise requires a microcentrifuge, 2 heating water baths, gel electrophoresis equipment, power supplies, horizontal gel boxes, trays, and cones. 1 or more thermocyclers with sufficient capacity to accommodate the number of student groups will be used, as well as an ultraviolet light source with an ultraviolet protective shield is needed to visualize the DNA brands.
There are 5 simple steps in order to conduct this experiment using the DNA in question. The first step is to generate complementary DNA, or cDNA, using RNA with the help of an enzyme called reverse transcriptase on a thermocycler for 60 minutes at 37 degrees Celsius. Then, the Rt-Pcr reactions are set up by adding a cocktail mix of Tacman, forward and reverse primers, Tacman probes, the cDNA you generated, and water into a microcentrifuge tube. And this tube is microcentrifuged briefly. Then, using a micropipette, 10 microliters of this is taken into a a well. The wells can range from 96 to 384 well plates. The plate is then sealed properly and centrifuged for 1 minute. This plate is taken into the Rt-Pcr instrument, where the instrument uses initial denaturation at 98 degrees Celsius for 5 minutes. Then the cycles begin. Denaturation at 98 for 30 seconds, annealing at 50 to 60 degrees Celsius for 30 seconds, and extension at 72 degrees for 45 seconds. Then final extension at 72 degrees happens. The data is collected at this point, and the Rt-Pcr instrument can give you a lot of results that you can then use to calculate your data. The real-time PCR machine that I mentioned earlier can help you gather enough data in order to collect the CT value and the PCR product. In this graph, we have the cycle number and the x-axis and the delta Rn in the y. Here we have the x0 and the xn. x0 is the initial copy number and xn is the PCR product after your number of cycles that you do. Here E is the amplification efficiency and n is the cycle number. Using this formula, you can get the initial copy number or the PCR product that you're trying to get. The CT value seen here is the point at which delta n crosses the threshold. CT is inversely correlated to the amount of nucleic acid in the sample, as seen by this formula. A lower CT value indicates high amounts of the target DNA. On the other hand, a higher CT value indicates that there's very little material for amplification, as seen here.
Some of the applications are listed here. We have agriculture and food industry. We also have gene expression, which includes southern blotting, DNA technology, and DNA sequencing. We have forensics, which PCR helps a lot in this field when using a very little segment of DNA, for example, a drop of blood or a single hair. And we also have human genetic testing, which will be the focus of our article in review.
An article from 2006 conducted an experiment about sex determination using PCR in order to identify the sex of an unknown donor. DNA samples can be compared to known samples or to DNA standards. You can amplify the X chromosome or the Y chromosome and separate these products and put them in the gel to identify the sex. As we know, females possess only an X chromosome, and therefore a band of 977 base pairs is observed. On the other hand, males possess an X and a Y chromosome and a band of 977 base pairs and a band of 788 base pairs is observed. PCR products from the DNA of female DNA can be observed here in this graph in line 1. As you can see, only the X chromosome is observed. Lane 2 is a male, the X and the Y chromosomes are observed. Lane 3 is a control. Lane 4 is a standard. And lane 5 is an unknown donor. So as we can see, lane 5 is a male.